Hello, my name is Dr. Amir Krivoy from Geha Mental Health Center and Tel Aviv University in Israel. In this presentation, I will give a short and concise overview of the main neurotransmitter relevant to psychiatry, beside dopamine, which was discussed in a separate talk. We will begin with the monoamines. The monoamine neurotransmitters family include serotonin, dopamine, histamine, adrenaline, and noradrenaline. This neurotransmitters group has been implicated in the pathophysiology and treatment of a wide variety of neuropsychiatric disorder. Monoamines are synthesized within neurons from common amino acid precursors and taken up into synaptic vesicles by way of a vesicular monoamine transporter. While they share metabolic pathways, they act differently on target cells by binding to specific surface membrane receptors. We shall now discuss the metabolism of monoamines. Monoamines shares common metabolic and catabolic pathways. Two enzymes that play major roles in the degradation of monoamines are monoamine oxidase, or MAO, and catechol O-methyltransferase, or COMT. MAO is located on the outer membrane of the mitochondria and oxidatively deaminates amines to their corresponding aldehydes. Two MAO isoenzymes with differing substrate specificities have been identified. MAO type A, which preferentially deaminates serotonin and noradrenaline, and MAO type B, which deaminates dopamine and histamine. Neurons contain both the MAO isoforms. Catechol O-methyltransferase, or COMT, is located in the cytoplasm and is widely distributed throughout the brain and peripheral tissues. It has a wide substrate specificity, catalyzing the transfer of methyl groups from S-adenosylmethionine to M-hydroxyl group of most catechol compounds. The catecholamine metabolites produced by this and other enzymes are frequently measured as indicators of the activity of catecholaminergic system. In humans, the predominant metabolite of dopamine and noradrenaline are homovanillic acid and 3-methoxy-4-hydroxyphenylglycol, MHPG, respectively. Serotonin metabolism is based on the amino acid tryptophan. Usually, tryptophan concentration is the rate limiting in serotonin synthesis. After tryptophan hydroxylation, 5-hydroxytryptophan is rapidly decarboxylated by aromatic amino acid decarboxylase, an enzyme also involved in dopamine synthesis to form serotonin. Catabolism of serotonin is mediated by the enzyme monoamine oxidase type A, which oxidizes the amino group to form an aldehyde. The elevation of serotonin levels by MAO inhibitors is believed to underlie the antidepressant efficacy of these drugs. After oxidation by MAO-A, the resulting aldehyde is further oxidized to 5-hydroxyindolacetic acid, 5-HIAA. Levels of 5-HIAA are often measured as a correlate of serotoninergic system activity. Histamine metabolism. Histamine does not readily cross the blood-brain barrier. Therefore, it is believed that histamine is synthesized within the brain, where it is formed by the decarboxylation of the amino acid histidine by specific L-histidine decarboxylase. Histamine is metabolized in the brain by histamine N-methyltransferase, producing methylhistamine. In turn, methylhistamine undergoes oxidative deamination by MAO-B. Adrenaline metabolism. In noradrenergic and adrenergic neurons, dopamine is actively transported into storage vesicles, where it is oxidized by dopamine beta-hydroxylase to form noradrenaline. The monoaminergic level and activity at the synapse is regulated by membrane transporters, which mediate the reuptake of synaptically released monoamines into the presynaptic terminal. Monoamine reuptake is an important mechanism for limiting the extent and duration of activation of monoaminergic receptors. Reuptake is also a primary mechanism for replenishing terminal monoamine neurotransmitter stores. Moreover, transporters serve as a molecular targets for a number of antidepressant drugs and psychostimulants. Specific transporter molecules for serotonin, dopamine, and norepinephrine have been well characterized. Within the presynaptic neuron terminal, the vesicular transporters serve to concentrate and store monoamines within synaptic vesicles. The transport and package of monoamines in vesicles may serve several purposes. First, to enable the regulated release of transmitter under appropriate physiological stimulation. Secondly, to protect monoamines from degradation by MAO. And last, to protect neurons from the toxic effects of free radicals produced by the oxidation of cytoplasmic monoamines. In contrast with the differential plasma membrane transporters for the different monoamines, a single type of vesicular monoamine transporter is believed to mediate the uptake of monoamines into synaptic vesicles within the brain. Therefore, blockade of this vesicular monoamine transporter by drugs such as reserpine has been found to deplete brain levels of serotonin, noradrenaline, and dopamine, and is associated with depressive reaction and increase of the risk of suicide and affective dysfunction. We will now review specific monoamines, neural circuits, and receptorial system of the different monoamines. Serotonin is generated in the raphe nuclei in the brainstem, which projects to the thalamus, basal ganglia, cerebellum, and the cortex. The cell bodies of these serotoninergic neurons are clustered in the midline raphe nuclei of the brainstem. The rostral raphe nuclei send ascending axonal projection throughout the brain, whereas the descending caudal raphe nuclei send projection into the medulla, cerebellum, and spinal cord. So far, there are 14 different known serotonin receptors. The 5-HT1 receptors comprised the largest serotonin receptor subfamily with 5 designated human subtypes. The most intensively studied of these has been the 5-HT1A receptor. This subtype is found on postsynaptic membranes of forebrain neurons, primarily in the hippocampus, cortex, and septum, and on serotoninergic neurons, where it functions as an inhibitory somatodendritic autoreceptor. There is significant interest in the 5-HT1A receptor as a modulator of both anxiety and depression. The antidepressant effect of chronic administration of serotonin reuptake inhibitors, or SSRIs, has been associated with downward regulation of 5-HT1A autoreceptors and may produce some behavioral effects via increases in hippocampal neurogenesis, which is mediated by postsynaptic 5-HT1A receptor activation. 5-HT2A and C receptors gained attention for being linked to the actions of atypical antipsychotic drugs such as clozapine, risperidone, or olanzapine. Analysis of the receptor binding properties of these drugs has led to the hypothesis that 5-HT2A receptor blockade correlates with the therapeutic effectiveness of atypical antipsychotics. The 5-HT2C receptor is expressed at high levels in many CNS regions, including the hippocampal formation, prefrontal cortex, amygdala, striatum, hypothalamus, and choroid plexus. Stimulation of 5-HT2C receptors has been proposed to produce anxiogenic effects as well as anorectic effects, which may result from interaction with the hypothalamic melanocortin and leptin pathways. 5-HT2C receptors may also play a role in the weight gain and development of type 2 diabetes mellitus associated with atypical antipsychotic treatment. Conversely, hallucinogens such as lysergic acid diethylamide or LSD display agonist activity at 5-HT2 and other serotonin receptor subtypes. Histamine. Most of histamine is located in mast cell and enterochromaffin cells, and only small portion is within the neurons. Histaminergic cell bodies are located within a region of the posterior hypothalamus known as the tubero-mammillary nucleus. Histaminergic fibers project diffusely throughout the brain and spinal cord. Ventral ascending projection course through the medial forebrain bundle and then innervate the hypothalamus, diagonal band septum, and olfactory bulb. Dorsal ascending projection innervate the thalamus, hippocampus, amygdala, and rostral forebrain. Interestingly, histaminergic fibers have varicosities that are not classical synapses. So histamine has been proposed to act as a distance from its site of release, like a local hormone. The hypothalamus receives the dentist histaminergic intervention consistent with a role for this transmitter in the regulation of autonomic and neuroendocrine processes. The brain histaminergic systems have been proposed to modulate arousal, wakefulness, feeding behavior, and neuroendocrine responsiveness. 4 histaminergic receptor subtypes have been identified and termed H1, H2, H3, and H4. The H4 was identified recently and is detected predominantly in the periphery. The other 3 histamine receptors have prominent expression in the CNS. H1 receptors are widely distributed within the CNS, with particularly high levels in the thalamus, cortex, and cerebellum. H1 receptor activation is associated with increased excitatory neuronal response. These receptors are the targets of classical antihistaminergic agents used in the treatment of allergic rhinitis and conjunctivitis. The sedative effects of these compounds have been attributed to their action in the CNS and have been associated with H1 antagonism, affecting the regulation of arousal and the sleep-wake cycle. The sedation and weight gain produced by a number of antipsychotic and antidepressant drugs have also been attributed to H1 receptor antagonism. The H2 receptor is highly expressed in various cells and tissues such as B cells, T cells, dendritic cells, gastric parietal cells, smooth muscle cells, and the brain and cardiac tissues. The H3 receptor is coupled to G protein and exclusively expressed in neurons. It is important for homeostatic regulation of energy levels, sleep-wake cycle, cognition, and inflammation. And lastly, the H4 receptor is coupled to G proteins and is expressed on a variety of immune cells as well as on other cells such as the spleen, intestinal epithelia, sensory neuron, and cancer cells. H4 receptors mediate the pro-inflammatory responses of histamine in both autocrine and paracrine manners. Noradrenaline. Noradrenaline-producing neurons are found within the brain, in the pons, and in the medulla in two major clusterings, the locus coeruleus and the lateral tegmental noradrenergic nuclei. The locus coeruleus is found in the dorsal portion of the caudal pons and contains approximately 12,000 tightly packed neurons on each side of the brain. These cells provide the major noradrenergic projection to the neocortex, hippocampus, thalamus, and midbrain tectum. The function of alpha receptor subtypes have been difficult to determine due to a lack of selective agonist and antagonist. Alpha-2 receptors display both presynaptic autoreceptor and postsynaptic action and all appear to inhibit cyclic AMP formation and to activate potassium channels with resultant membrane hyperpolarization. These receptors regulate neurotransmitter release from peripheral sympathetic nerve endings. Within the brain, the stimulation of alpha-2 autoreceptors inhibits firing of the noradrenergic neurons of the locus coeruleus, which have been implicated in arousal states. This mechanism has been proposed to underlie the sedative effect of the alpha-2 receptor agonist clonidine. In addition, the stimulation of brainstem alpha-2 receptors has been proposed to reduce sympathetic and to augment parasympathetic nervous system activity. This action may relate to the utility of clonidine in lowering blood pressure and in suppressing the sympathetic hyperactivity associated with opiate withdrawal. Activation of alpha-2 receptors inhibits the activity of serotonin neurons of the dorsal raphe nucleus, whereas activation of local alpha-1 receptors stimulates the activity of these neurons, and this is thought to be the major activating input to the serotoninergic system. Acetyl-choline. Acetyl-choline is synthesized by the transfer of the acetyl group from acetyl coenzyme A to choline in a reaction mediated by the enzyme choline acetyltransferase. Two large clusters of cholinergic projection neurons are found within the brain. The basal forebrain complex, which consists of cholinergic neurons within the nucleus basalis of Maynard, the horizontal vertical diagonal bands of Broca, and the medial septal nucleus, and the mesopontine complex. Choline is taken up into cholinergic neurons by a high-affinity active transport mechanism, and this uptake is the rate-limiting step in acetylcholine synthesis. After synthesis, acetylcholine is stored in synaptic vesicles through the action of a vesicular acetylcholine transporter. After vesicular release, acetylcholine is rapidly broken down by hydrolysis by the enzyme acetylcholine esterase, located in the synaptic cleft. Much of the choline produced by this hydrolysis is then taken back into the presynaptic terminal via the choline transporter. Acetylcholine receptors. The muscarinic receptors M1 are the most abundantly expressed muscarinic receptors in the forebrain. Pharmacological evidence has suggested their involvement in memory and synaptic plasticity. M1 receptors are the most abundantly expressed muscarinic receptors in the forebrain. Pharmacological evidence has suggested their involvement in memory and synaptic plasticity. M1 is also common in neurons innervating smooth muscle. The M2 muscarinic receptors are located in the heart, where they act to slow the heart rate down to normal sinus rhythm by slowing the speed of depolarization. M3 receptors are G-coupled and mediate an increase in intracellular calcium. They typically cause contraction of smooth muscle. such as that observed during bronchoconstriction and bladder voiding. M4 receptors function as inhibitory autoreceptors for acetylcholine. Activation of M4 receptors inhibits acetylcholine release in the striatum. Therefore, antagonists at that site might induce clinical features of cholinergic activation. The location of M5 receptors is not well known. Nicotinic receptors have been implicated in cognitive function, especially working memory, attention, and processing speed. Cortical and hippocampal nicotinic acetylcholine receptors appear to be significantly decreased in Alzheimer's disease, and nicotine administration improves attention deficits in some patients. The alpha-7 nicotinic acetylcholine receptor subtype has been implicated as one of many possible susceptibility genes for schizophrenia, with lower levels of this receptor being associated with impaired sensory gating. The reinforcing properties of tobacco use are proposed to involve the stimulation of nicotinic acetylcholine receptors located in mesolimbic dopaminergic reward pathways. Gamma-aminobutyric acid, or GABA, is the major inhibitory neurotransmitter in the brain. GABAergic neurotransmission has been implicated in a broad range of neuropsychiatric disorders, including anxiety disorders, schizophrenia, alcohol dependence, and seizure disorders. GABA derives from glutamate, the major excitatory neurotransmitter, by the removal of a single carboxyl group by glutamic acid decarboxylase, also known as GAD or GAD, using vitamin B6 as a cofactor. In the CNS, the expression of GAD is restricted to GABAergic neurons, although in the periphery it is expressed in pancreatic islet cells. Two isoforms of the enzyme were identified. GAD65 is localized to nerve terminals, where it is responsible for synthesizing GABA that is concentrated in the synaptic vesicles, whereas GAD67 or GAD67 appears to be the primary source of neuronal GABA. GABA is catabolized by GABA transaminase to yield succinic semialdehyde. GABA transaminase is a cell surface membrane-bound enzyme expressed by neurons and glia. Drugs that inhibit the catabolism of GABA have anticonvulsant properties. For example, one of the mechanisms of action of valproic acid is the competitive inhibition of GABA transaminase. The synaptic action of GABA is also terminated by high-affinity transfer back into the presynaptic terminal as well as into astrocytes. The active transport is driven by the sodium gradient so that upon depolarization, transportation of GABA out of the neuron is favored. GABA transported into astrocytes is catabolized by GABA transaminase and ultimately converted to glutamate and then to glutamine, which is transported back into the presynaptic terminal for GABA synthesis. Transamination generally occurs where the parent compound alpha-ketoglutarate is present to receive the amino group, thereby regenerating glutamic acid. The GABA-A complex, when activated, mediates an increase in membrane conductance with an equilibrium potential near the resting membrane potential of -70 millivolts. This typically results with an influx of chloride, causing membrane hyperpolarization. Hyperpolarization is inhibitory because it increases the threshold for generating an action potential in the membrane. The receptor channel complex is heteropentamer. The GABA binding site is at the interface of the alpha and beta subunits. The benzodiazepine binding site is at the interface between the gamma and the alpha subunits. Alpha-1 subunit plays a major role in the sedative and amnestic effects of benzodiazepine, whereas inactivating the benzodiazepine site on the alpha-2 subunit eliminates the anxiolytic effect of benzodiazepine. Barbiturates allosterically increase the affinities of the binding sites for GABA and benzodiazepine. Other compounds may act at a distinctly different site. By that, they allosterically enhance agonist ligand binding to the receptor and increase the duration of chloride channel opening. GABA-B receptors are widely distributed throughout the nervous system and are localized both presynaptically and postsynaptically. The postsynaptic GABA-B receptor causes a long-lasting hyperpolarization by activating potassium channels, while presynaptically they act as autoreceptors and heteroreceptors to inhibit neurotransmitter release. GABAergic dysfunction has been associated with anxiety disorders, especially panic disorder, as well as with major depressive disorder. Glutamate. Glutamate is an amino acid first synthesized in 1896 from gluten and therefore has a distinct taste. It is the most prevalent excitatory neurotransmitter in the brain. Glutamate can be synthesized from several biochemical sources. Glutaminase converts glutamine to glutamate in the brain. It is the reverse reaction of glutamine synthase. Glutaminate oxaloacetate transaminase enzymes convert alpha-ketoglutarate to glutamate. The reaction also generates oxaloacetate. Glutaminate oxaloacetate transaminase catabolizes the reversible transference of aspartate amino acid group to alpha-ketoglutarate and produces glutamate and oxaloacetate. Specialized astrocytes surround the glutamatergic synapses. The astrocytes express the two sodium-dependent glutamate transporters that play the primary role in removing glutamate from the synapse, thereby terminating its action. EAAT1 and EAAT2, the excitatory amino acid transporter. The neuronal glutamate transporter EAAT3 is expressed in upper motor neurons, whereas EAAT4 is expressed primarily in cerebellar Purkinje cells. Three families of ionotropic glutamate receptors have been identified on the basis of selective activation by synthetic analogs of glutamate. These include alpha-amino-3-hydroxy 5-methyl-4-hydroxazole propionic acid, or AMPA, kainic acid, or kainate, and the N-Methyl-d-Aspartate acid, or NMDA receptors. They all activate a membrane ion channel. The metabotrophic glutamate receptor are so designated because their effects are mediated by G proteins. All mGluRs are activated by glutamate, although the sensitivities vary remarkably. To date, 8 mGluRs have been cloned. The NMDA receptor has been heavily investigated. It is composed of two units which form a membrane calcium ion channel. Upon depolarization of the membrane and a removal of a magnesium ion which blocks the channel, and activation of glycine and glutamate sites of the receptor, the channel opens. The receptor was found to be involved in processes associated with long-term potentiation, memory, and learning. The theory that hypofunction of NMDA receptors is an etiologic factor in schizophrenia initially arose from the observation that phencyclidine, or PCP, and related dissociative anesthetics that block NMDA receptors produce a syndrome that can be indistinguishable from schizophrenia. Dissociative anesthetics are so named because they prevent the acquisition of new memories while the patient is apparently conscious. To conclude, in this short overview, we've covered the introduction to the main known neurotransmitter relevant to mental disorder and behavior. Thank you for listening.